Hi, this is Mrs. Adcock, and you are having Science with Adcock. Last video I made was about replication, and today I'm going to go over transcription. Okay, let me turn you over here. All right, so what we have here is my nice little cell made up here. I have my nucleus. Inside my nucleus, I have my DNA, which we know is the genetic makeup, okay, of our bodies. And then my cell membrane, which is out here, and then my cytoplasm in here. Yes, I am missing many organelles. In fact, I'm missing all my organelles. I do not have ribosomes or Golgi apparatus, endoplasmic reticulum. I get it. But we are simply doing transcription today. And what that is, and we're going to break down this word. Okay, so trans, the root word, this prefix means across, script, and then we're going to add in shun, and that means to write. So we are going to write across, all right? Basically, we're taking the message of DNA and we're just making it small enough to exit the nucleus. So we have a message here. DNA is so large, it's so large it just cannot pass through the nuclear membrane. So what it has to do is it needs to get small enough to then pass through the nuclear membrane to then be able to translate that into a protein. Okay, so how do we do this? All right, so we're going to focus on this step here. We are going to focus from here to here. All right, so these simple steps from here to here, this is our transcription. Now remember that DNA has, wait a minute, let me just get this down. DNA has it uses 4 letters of the alphabet, okay? It uses A, T, C, G. And let me see, I'm gonna add one more, we'll make that an A. So here is my 5' to 3' end of DNA. Let me move this a little bit closer here, okay? And if I were to do my complementary strand, I know that it has to be 3'. Remember, that was the position of our sugar, our 3' or 5' end. And it would look like this. Okay, so if I did my complementary strand of DNA, that's what this looks like. Now, understand again, DNA is so large, it's microscopic. We can't see it, without the aid of electron microscopes and, ways to make this extremely visible. But in a microscopic sense, it's too large to exit the nucleus. So transcription is to write this message in a way that it can leave the membrane, leave the nucleus, pass through the membrane, and then continue what DNA, what our genetic makeup or message is telling us to do. So, I talk about proteins. Our body needs to make proteins. Like what? Well, right now we're in a world where corona is rampant. And, if for some reason the antigen corona enters your body wants to make antibodies to fight off the antigen corona. So again, your body's message would be, make a protein, make this antibody. There's something in there that shouldn't be there. Let's fight it off. And so that's one thing. That's one way to look at it, a type of protein that your body has to make. All right, so how does this happen? Now we've covered replication, so we know how to make more copies of DNA. We do that again, why? We're growing, right? You're a child and you grow. Well, your body makes more cells. We do that via mitosis, and we have to replicate our DNA. You injure yourself, you need to replace tissue, all right, or repair damaged tissue, worn out tissue. We do that via mitosis, okay? So we've already replicated. We've made as many cells as we need. Now we need to get the message out to make proteins. And in the case I gave you is to make an antibody to fight the antigen. So here's DNA. And so we're actually going to make RNA's message. RNA is the short version of DNA. Again, we are writing this message in kind of in a cross format of going right across letter, base to base, and we're writing that. And so which strand do we use? We have two strands here, right? I have my leading and lagging strand. This goes back again to replication. I'm actually going to focus on my leading strand. All right, so I'm going to use this strand here, and this is the one that I would be reading the message from. So remember, if I'm using my leading strand, and this is 5', I know that RNA, its version, right, of this short message is going to be 3 to 5'. So again, we add on 5 to 3. We do this in the same way that you were doing replication because leading strand was continuous, whereas the lagging strand did just that. It lagged. It was discontinuous. We needed ligase. We had Okazaki fragments. There was much more going on. So it was more complicated. But here, again, we're going to use the leading strand. So we have a nice flow of bases coming in to read the message. We're going to go across DNA. We're going to read, and we're going to write that down. Okay. So here's the thing, though. This is what's interesting, is we no longer use thymine. DNA uses 4 letters of the alphabet: A, T, C, G. RNA uses A, U, C, G. We no longer use thymine. We now use what is called uracil. Okay, that's that guy right there. By changing that base pair as well, we're now making the message even more short. All right, we're shortening it even more. We're making it even, so small enough that eventually we can exit the nucleus. And that's the goal, because we want to get here. Okay. All right. So I'm going to transcribe, and I'm going to write what matches across here. Well, T always pairs with A. Normally I would write a T, but that is not what is, complementary to this. So I'm going to write U because this is RNA. Remember, when you see a U, you know this is RNA. I'm going to keep going ahead. Remember, again, can't write T, we would write U. And also what you'll notice is RNA strand is very similar to what we call the lagging strand of DNA. So you'll notice this is almost identical. The difference, though, is this base right here. This is now uracil instead of thymine. Okay, so we've done exactly what we just wanted to do. We transcribed, we wrote the message, and so now, I'm going to change this a little bit, though. So now, I'm going to use that color just so that is more clear. We took the message, the double-stranded message, and we just made it into something smaller. Well, here's the thing. Right now, this is a pre-strand of messenger RNA. Okay, so pre-mRNA. Why? Well, let me erase this here. So we have something that looks like, let's say, it looks like this. Hopefully the color, yep, you can see the color is blue and so forth. And so we have the blue here and blue here. There are two parts to my pre-mRNA. We have introns and exons. Okay, so we have introns and exons. Introns, again, this is your DNA. So this is your message. This is your genetic makeup. But when we're making proteins, there's information in here that is just irrelevant to the job at hand. And so I actually use that as a way to remember that introns are irrelevant to what's happening. So again, focus on the I and the I. Maybe that's a good way for you to remember this. The introns are irrelevant for the message of that protein to be made. So what happens is your body will cut out, again, not chopping, but your body will actually remove from the pre-mRNA strand. It will remove that, and you will wind up with mRNA. So here's going to be my pre-mRNA strand, and this would be my RNA strand. And notice again, just visually looking, it's shorter. So we've taken a huge message here, a double-stranded message. We made a pre-mRNA strand, and then we even made it shorter. We spliced out, right? We spliced out what was not necessary. Okay, and let me get rid of this here so there's, again, no confusion there. And this guy right here is our official messenger RNA. And this becomes now the final RNA strand ready to exit. Okay? Today is just about transcription. We will talk about translation, but today is only transcription, and it's the first step in protein synthesis. Hopefully this made it easier for you, and let me know what you think. So comment, like, and subscribe, and I will talk to you next time. Bye.